All living organisms start from a single cell, yet they grow and develop into complex systems made up of many specialized cell types. This transformation from a single, undifferentiated cell into a multicellular organism with diverse cell types is a process called cell differentiation. Today, we'll explore how cell specialization happens, why it's essential for life, and the critical role that environmental factors play in shaping this process. Let's begin with the foundation. What is cell differentiation? Cell differentiation is the process by which unspecialized cells, like stem cells, develop into specialized cells with distinct structures and functions. For example, a single fertilized egg can give rise to all the different cell types in an organism: muscle cells, nerve cells, blood cells, and more. Each of these cells has a specific role, making differentiation crucial for the growth, development, and survival of multicellular organisms. The process of differentiation is guided by the organism's genetic instructions encoded in DNA. Every cell in an organism contains the same DNA, but not all genes are active in every cell. Differentiation occurs when certain genes are turned on or off in a controlled way. This selective gene expression determines the structure and function of each cell. Imagine a developing human embryo. In its earliest stages, it consists of identical cells. These cells begin to receive chemical signals that activate specific genes. Some cells might start producing proteins needed for muscle contraction, while others begin forming components of the nervous system. Over time, these changes make each cell type uniquely suited to its role in the body. A key player in this process is stem cells. Stem cells are undifferentiated cells with the unique ability to either remain in their unspecialized state or become specialized. They are categorized into different types based on their potential. Embryonic stem cells, for instance, can differentiate into almost any cell type, while adult stem cells are more limited, often giving rise to cells within a specific tissue like blood or skin. But cell differentiation is not just a one-way journey. Some cells can de-differentiate or return to an unspecialized state under certain conditions. Scientists are exploring this capability for medical purposes, such as regenerative therapies, which aim to replace damaged tissues with lab-grown cells. Now let's turn to the role of environmental factors in cell differentiation. While genes provide the blueprint for differentiation, the environment acts as the architect, influencing how and when genes are expressed. One of the most significant environmental factors is chemical signaling. During development, cells communicate using chemical signals like hormones and growth factors. These signals guide cells to adopt specific fates. For example, in a developing plant, the hormone auxin determines which cells become root tissue and which form shoots. Similarly, in animals, chemical gradients play a pivotal role. Consider the early stages of human development. The cells in an embryo are exposed to varying concentrations of signaling molecules depending on their position. These gradients provide instructions that help cells specialize into different tissues, such as skin, muscle, or brain. Another critical factor is physical interactions. Cells in a growing organism are surrounded by an extracellular matrix, a network of proteins and molecules that provide structural support. The stiffness, texture, and composition of this matrix can influence cell differentiation. For instance, stem cells grown on a rigid surface in a lab are more likely to differentiate into bone cells, while those on a softer surface may become nerve cells. Temperature and light also impact cell differentiation, particularly in plants. In many species, exposure to light triggers the differentiation of cells into chloroplast-rich tissues that perform photosynthesis. Similarly, temperature changes can influence which genes are active, affecting how cells develop. For example, certain reptiles exhibit temperature-dependent sex determination, where the ambient temperature during embryonic development determines whether an organism develops male or female reproductive cells. Nutritional availability is another key environmental factor. Nutrients provide the building blocks and energy required for cell growth and differentiation. For instance, A deficiency in certain vitamins or minerals during development can lead to impaired differentiation and result in structural or functional abnormalities. External stressors, such as toxins or pollutants, can also alter cell differentiation. These factors might disrupt normal signaling pathways, leading to abnormalities or diseases. Understanding how environmental stress affects cell differentiation is critical for both developmental biology and health sciences. But differentiation isn't limited to the embryonic stage. It continues throughout an organism's life, especially in areas where tissues are constantly renewed. Consider your skin. The outermost layer is made of specialized cells called keratinocytes. These cells originate from undifferentiated stem cells in the deeper layers of your skin and differentiate as they migrate toward the surface, where they form a tough, protective barrier. Cell differentiation also plays a crucial role in healing. When you get a cut, undifferentiated cells in the area are activated and begin to divide and specialize to replace damaged tissue. This remarkable ability highlights the body's reliance on differentiation for maintaining health. However, errors in the differentiation process can lead to serious consequences. If cells fail to differentiate properly, they might become dysfunctional or develop into abnormal growths, such as tumors. Cancer, for instance, often arises when cells lose their ability to specialize and begin dividing uncontrollably. To conclude, cell differentiation is a fundamental biological process that transforms unspecialized cells into specialized ones, enabling organisms to grow, develop, and maintain themselves. While genes provide the instructions, environmental factors such as chemical signals, physical forces, and external conditions shape how differentiation unfolds. This intricate interplay between genetics and the environment ensures the diversity of cell types needed for life. By understanding how cell differentiation works, scientists can uncover new ways to treat injuries, regenerate tissues, and combat diseases, offering hope for advances in medicine and biotechnology.